Welcome to the start of the after coffee break session. The next speaker will be Ruiz Guzman, who is talking about large language model-based framework to orchestrate and manage analytical pipelines on multidimensional remote sensing dataset. So please welcome Ruiz Guzman.
Hello, everyone. It's been an exciting week learning about new technologies, new devices, new sensors for pretty much be able to map every single part of their crops. But today I'm going to focus on a very specific aspect, and it's about how we can actually integrate all of this set of technologies, all of this output that these models, algorithms that we trained to improve decision-making capabilities. And I would like to start asking the audience if you probably, who here have been using ChatGPT for writing or improving the coding skill? Someone? Well, good. And the fascinating thing about this is that how this new AI foundation model are moving really quickly. Indeed, we are in the phase that we are moving from single task models, single task algorithms, to those sophisticated foundation models that they can actually understand multimodal data in the same kind of way that human does. The reason why we can reason is because we can actually understand, we can actually understand multimodal data and make decisions in real time. Yeah, I don't know why doesn't show the first one, but anyway, so we work for that. We are working on Texas A&M AgriLife Research. It's part of the Texas A&M University system. So we are around 13 research stations centered across all the state. We are specifically located almost in Mexico, the Rio Grande Valley, and we have different areas of research. Us specifically, our lab, we are focusing on innovative new technologies for agriculture. We are trying to kind of collaborate with research, different universities, different organizations across the globe to come with different technology that can help us to improve our research practices. So specifically what we want is that we have developed a really powerful platform capabilities to map our crops, collect information with different sensors like UAV, ground penetrating radar for root phenotyping. And now we are focusing in how we can actually collect all of that information that come from different toys, different fancy technology that extract information from the electromagnetic spectrum and how we can transform that information into pictures. That means that what we're going to learn about this new framework called LegoPy is that how we are using these foundation models to improve in the way farmers, research can access to all of this information that we are daily generating. So, and I want to emphasize that what we are trying to do here is we want to transform this non-structured data, like we call it the observations or measurements, and how we can transform that into a structured data that eventually we can use for downstream analysis, like machine learning, all of these fancy models that we have today. So, and something that we realize is that high throughput phenotypic pipelines are data intense. We have all of these platforms to collect data through all of the crop cycle. And there is a lot of kind of challenge with that, One is that we have this structured data, different for such as tables, databases, crop data sets, so on. But we also have this non-structured data set that, like genome sequence data, images, hyperspectral data, remote and proximal sensing data that we generate. So it's a really tight process. Data analysis in remote sensing is complex, especially because here is one example. How many tools you need to have in order to transform your flights in something that you can potentially use or breeders can use to do inference based on that data? How many software you need to play around, you need to be expert to be able to kind of extract features from that data and then be able to kind of give the information to breeders and research that they really need. So we have done a lot of research in terms of integrating UAV platform and ground penetrating radar, specifically for above ground and below ground biomass estimations. And one of the limitations that we found with the GPR world is like, even though there is a lot of people effective in this kind of technology, well, we were able to make it work. And the way that we did it is like, we needed to come with our own software. So we developed GPR Studio, and GPR Studio is an ecosystem of Python libraries and software working together to transform the GPR data into information and fixture that we can use for good biomass estimation. And it worked pretty well after like a lot of research, years of like working on that. This is the software. You have like a visualization software, but you also have a Python API that you can actually use to interact with your data. You can transform that data in more sophisticated algorithms that you can use for prediction and so on. But the limitation was that, this tool has been developed for geophysicists. So how breeders can actually use this tool for biomass or root biomass estimation? Well, we needed to develop our own software, because if you realize in the ecosystem of tools that these companies provide to you, you cannot go further of these buttons and options that you have in the UI. That means that we come with this software. So we recently were able to integrate LiDAR data and GPR data for reconstruct 3D above ground and below ground biomass estimation. We recently submitted a paper, this is potato here, where we were able to transform the GPR signal to a 3D representation of the tubers. But today talks not about these data fusion approaches or sensors. We're gonna focus in this, One of the limitations that we have so far with this technology is that there is a new sensor every day. There is a new software every day for GPR, and even like it was mentioned before, there are people working the same thing, and you don't realize that we are working the same stuff. And also, something interesting is that observations are biased to the interpretation. You're good at collect UAV images, and got like this pretty picture that can tell you a lot about your crop. But when you transfer that data in something that the breeders and pathology they can interpret, you're gonna find out that they have different point of view, and this can be a disease or like a pathogen for someone, like it's a plant pathologist, but it could represent something related with oil soil quality or something like that. So the question here is how you can integrate all of this information that you derive from this multi-sensor platform, but incorporating also expert domain knowledge that eventually is going to help you to kind of bring all of this information in context and improve the decision capability that you have in your program. And this is why we developed LegoPy. LegoPy is a framework. It's actually like general purpose framework. It's a crop agnostic framework that it has been focused specifically in managing and optimizing workflows for analyzing and processing multi-data in agriculture application. But this can be going beyond that, because we're gonna see how this LegoPy works. So the foundation of LegoPy is that recent advances in LLMs have demonstrated that these models can actually reason. So that means that it has changed in the way how we interact with these models today. So the key idea about that is LLMs, large language models, are these sophisticated models based on transformer architecture that they have been specifically trained to understand human languages. They are really extremely good to transform language into a structured data that you can use to orchestrate your pipelines. And the foundation behind of that is they are autoregressive. The autoregressive design of this model enables them that they generate one token at the time, but they are able to get back and use the previous token and adjust the output. This is extremely useful because if you provide the model with intermediate steps how to solve problems, they're gonna be able to learn how you can actually improve how you can actually not only just generate the next token, but provide with the reasoning steps that you need to do to solve a specific task. So this change how we actually interact with large language model today. This is a standalone interaction with LLM. You just go open ChatGPT and you ask, what is 1 plus 2? And just generate the next token. But what about if you ask this question? Which crop, cotton or soybean, is better suited for the hot and dry climate in West Texas? So probably these LLMs haven't been trained on so much information on plant breeding or agricultural practice or so on, right? But today, there are different design patterns that you can actually provide these models with external information and data that they can incorporate into the context. So that means that the output is not going to be specifically based on the data that they memorize during training, but they're going to also use the information that you provide in the context. And we are talking about that these models can actually handle up to like 2 million tokens. 2 million tokens is almost like 9 Harry Potter books together. Imagine how much information you can feed into this model. Another kind of interaction that you're probably going to need to have is like, what about if I have a model, like soil model or weather model that you train in your data? And you want the model be able to access that model to bring context into the capability. So we can also do that. If I have a specific function and a script that run in the backend a model that you have been training your data, you can actually make the model be aware about that. That means that the next time he's gonna, I cannot execute code, but I know that there is a function that I can potentially use in the backend, and it's gonna give me the weather prediction or whatever you guys can think. But there are even more sophisticated applications of LLMs today. For instance, those where reasoning is required. Those complex problems that cannot be solved with a single interaction, but the model needs to actually get back and think about it and then come with an approach to solve the problem. The unified solution that we have today in the state of the art LLMs is agents. Agents extend beyond the text generation, which is what ChatGPT does, and incorporate advanced cognitive functions such as reasoning, planning, and decision making. And the way this actually works is that you train a model to be able to reason and make decisions. And then this model can have memory. That memory can be all the paper that you published last year when it's all the overview or the review of the agricultural practice that you have been building in your reading program or so on. And they can use all of that information in the memory to address any question that the user can potentially ask. So by harnessing advanced capabilities in our language models, they can autonomously execute and solve problems. This is an example. Imagine that the farmer asks, what is the weather today in San Francisco? For sure, the LLM is not trained with real-time data. But if you provide in the context that there is one function, one model that you train to be able to bring information from the weather in real time, it can actually feed that into the context. So that means that the agent can grab this tool, execute weather, which could be a Python function, an ARP function, and then provide you with an update version of that. So this is how LegoPy actually works. So you can create your custom blocks. Think about your analysis as a set of steps, and each step could be a block. One step is gonna be load the model, load the data, and so on. Think about like a Lego, building a Lego. Probably if you guys have son, you're very familiar with this. So we're gonna have people that have expertise in genomics data analysis, LiDAR data analysis, JPR data analysis. So LegoPy provides with a LegoPy registry. The registry is, imagine a database of thousands of blocks that you can use. Well, we haven't gotten to that point, but we have developed some for remote sensing application and agriculture specific. So you can use those blocks, and you have different ways to put these blocks together. One is using a UI, an interface. One is if you have some Python skills, you can actually develop your own custom analysis. But also, if you want to the model be able to suggest what could be the potential pipeline that fit into your analysis, you can ask the LegoPy, and he's gonna try to figure it out based on those blocks and information that he has about them in the database, how he can arrange those blocks so that it can provide you a pipeline that you can potentially execute in your data. So LegoPy philosophy is centered on fostering collaboration among agents and humans to tackle intricate tasks. So this is one of the high-level components of LegoPy. You have the user interface, but you have the Python APIs that enable you to create the agents on the blocks. And you have 3 different engines. We know that handler data is an expensive task, especially in the remote sensing context. So we relied in tools such as Kubeflow, Docker, or RAID, which are the tools that have been taught to deploy high-performance pipelines data analysis. But we also have the reasoning engine. The reasoning engine is because we don't want a reader navigate through a very complex software that have 2,000 different buttons to do just generate the vegetation indices. That means that you can actually use natural language processing to talk with LegoPy, and he's gonna figure it out. Hey, I have this block. Just use this block. He's gonna create the pipeline. He's gonna execute for you and get the data back. And we have 2 different data storage. Metadata, when you can save metrics, prediction accuracies, or any structured metadata that you want to save. But you have also a storage artifact, which is focused on, imagine that you generate an orthomosaic or one of the agents in the backend, run one function that creates that orthomosaic, and then you need to some a kind of a storage to save that output there. So this is when we talk about a non-structured data. And we have also a RAG engine, because imagine that you want to submit a script that compute vegetation index. But there are 2,000 scripts that do the same thing. So that means that LegoPy internally use this RAG engine to identify that the code that you are trying to write, even if it's different to someone that somebody else wrote, is similar. That means that it's really kind of important to have another block that does the same thing. Another thing that you can do is you can put multiple LLMs or models to collaborate each other to solve more complex tasks. This is what we call in agents collaboration patterns. So for the short video of 1 minute that I'm just gonna move quickly so I can show the video. Well, another challenge that we have is that how we can incorporate information from different multimodalities and talk about images, cloud points, or this kind of stuff to the same data structure. Well, we are currently using XLRate, which is a format optimized specifically for this kind of scenarios, when you can have information about your plot, and this information can be multidimensional. So this is the example that I will show. Imagine that you are a breeder, and you don't have someone that is expert in your program that want to kind of help you to figure it out how to generate an orthomosaic, how to compute the vegetation index, save them in a CSV file, try to determine what is the correlation with yield, and how you can potentially use that information to improve predictions. Well, so you can make these kind of agents collaborations when each agent is gonna be doing something different. For instance, the remote sensing agent is gonna have access to different script or function that you have created in the backend to automate all of these kind of things. You have another growth physiologist that is gonna be a retrieval agent, where all the paper that you have written about yield, you can pass to the model, so that means that the model can bring that into the context. So at the end, LegoPy provides a high-level API to orchestrate, develop, implement, and deploy these kind of solutions. And just to provide a better picture of how this actually works, this is a 1-minute video that shows you everything working together. So you have 4 different agents here, remote sensing expert, So all of these agents is gonna be collaboratively working to solve a specific task. So the first question was about, go to my account in 2x and bring what projects I have in the access to. But you can also ask questions such as, can you please generate the vegetation index? So you're gonna have some kind of stuff in the backend that you're gonna generate the indices, and I'm gonna bring that into the context. So here is generate the indices for you. So then you can go to the genomic data analysis. Can you create a relationship matrix from my market data. And you're gonna return the information that you expect. And finally, you can have a crop expert that can combine all of this information to provide a better decision making. And so he's gonna provide here with very detailed explanation about what you can do to improve and how you can do for. So I'm short in time, so this is one of the possibilities, LegoPy facilitates multimodal data function integration, improves content-aware decision-making capability, elevate data interpretability and accessibility. Reproducibility is a good thing, flexibility and crop agnostic, facilitate prime deployment. And so at the end, so we are open to collaborate. So I want to extend my acknowledgement to the AI and the remote sensing team in the West Laco station. So we are open to collaborate. We are right now looking where LegoPy can fit in and how you guys can potentially use that into the breeder programs to improve decision making. And with that, thank you so much.
So thank you very much for the nice talk. Is there any question? Just there in the front here.
Hi, thanks for the presentation and an interesting approach to using the AI to also help with analysis tooling. One specific question about the analysis part of it. Do you have any way in place to keep track of which scripts and which tools the analysis pipeline that is chosen by the agents is recorded so that you have the computational provenance, like how actually were the results imputed?
Yeah, so indeed the way actually that works is that even though these models have the capability to generate code, they probably, because they haven't been fine-tuning the script that you have created for a long time, you can just bring all of them and you can actually specify which agent is going to have access to this particular function or blocks that you and the agent is just going to use, they're going to try to understand what you want to do, and they're going to figure it out internally that, oh, these functions can be addressed by a script that you wrote or something like that, That means that the agent internally, they are really good interpreting human language. So that means that they understand exactly what you mean, but they cannot execute code. But you can provide them with different tools so they can actually use in the backend to generate and bring the context information for generate the output that you expect. That means that you are free to provide them with the methods and the functions that you have implemented by your own.
Okay, so one very short question, just right here in the middle.
Is the code open source and available on GitHub? And very briefly, won't you have problems with such a generic and known name?
So we're going to open source the code. So we right now is we have a paper ready for submit. We are trying to think where can be a good journal to submit that paper. When once we submit it, we're going to make all the code available. And what is the other question that you asked? Legal. This is a really good point. This is something that actually right now we are working because we know that Lego, I just skipped that, but LegoPy is watermark. So the reason why we call Lego is because the idea is that you bring all of these blocks together to solve complex problems that the agents they're gonna use in the backend to solve the problem, but we're probably gonna change the name because this is a watermark name.
Okay, so thank you very much for the nice talk.
Thank you.
Thanks. So the next talk will have the acronym SMART. Love that one. It's Speedy Measurement of Arabidopsis Rossette Traits, and it's from Suxing Liu. So thank you very much.
Thank you. Thank you, Stephen. Welcome to the last talk of this long talk session. I'm Suxing, and I'm from University of Arizona. And today I'm going to talk about the side project I developed just out of interest. Later, how it becomes so interesting. And special thanks to give to my mentor, Alexander, who gave this software a very short and easy to remember name, SMART. In here, you already see the input image of the Arabidopsis, and on the other side, you will see the output of one of the result of SMART. What does the result mean? Let's find out. And from computer science, I'm in the computer science part, but I have been working in applied science for several years, and I have learned many cool facts. One of the cool facts was plants was able to sustain and feed themselves through the famous photosynthesis process. During the photosynthesis process, at the same time, they can reduce the actual CO2 in the air and bring them to the root, to the soil. And in here, in the photosynthesis process, the plant shows the color. Most of the plants show the green color. Why the plants show the green color? Because in the light, in the sunlight, there are many wavelengths. When it hit the surface of the plant, most of them was absorbed by the chlorophyll. This has done for a long time, by the research can confirm that, and only yellow and greenish was reflected by the surface. In here, this question was pretty old, but for me, I've been thinking from computer science part, I have noticed the black color was the most efficient one to absorb all the wavelengths. Why plant was not black? This is drawn my attention why the plant color was so important. So I really think about more to how to quantify the plant surface colors. Here I read more studies and excellent papers. Recently there was one paper trying to point out there are some connections between gene and the color part. They are published, their result in Nature, and they have been using many sophisticated cameras, like RGB camera, hyperspectral camera. They want to capture different wave and quantify the colors, and use the expensive pulse amplitude modulated fluorometer to capture the chlorophyll content. So I've been thinking, is there some way to just using RGB cameras or get the similar information, or at least get some good estimation? So this comes with question how we analyze the colors, how we quantify the colors. It sounds easy, but when we teach machine or computer to do that, it is not so easy like we thought. It's funny that many people using LEGO images. Here I give you the same examples in here. In these LEGO images, as a human, we can see that there are 4 kinds of different colors, despite the shadows, despite their small round shape, despite the edges. But if we teach the computers, they will plot all the colors, pixel by pixel, into the histogram. Then we can see there are thousands of different colors in this histogram, and we can't tell which one was important, which one was not important, and there's no clue to find out how we quantify the colors. So I introduced a dominant color clustering algorithm. This is a foundation and basic idea of SMART. It will be used as a very basic idea. And in here, we can see that we introduced the machine learning algorithm. In here, we try to use unsupervised machine learning without any human label or interfere to find out the dominant colors in this image. You can see on here, the LEGO images, we plot that or visualize them, all the colors in a 3D RGB space. We can see that there should be some centers, some clusters grouped together. So after that, we're using the dominant clusters and find out the centers. In the meantime, we also record the center values of all the clusters in this group. On the right side, you will see the LEGO images coming from the original one, go to the dominant color one. This is the simplified version of the color quantification. This is the whole process of dominant colors, and this is the basis of the SMART. But what is the usage of this dominant cluster if we want to analyze the plant surface colors. Here, we tried very boldly using the input of one of the test images. We can definitely segment the plant out, then use the same dominant colors clustering method to apply to the plant surface. In here, we can see that the SMART was able to quantify 3 levels of different green. In here, I can say carefully, it might be yellowish. So the output of SMART will tell you that maybe there are 42% yellow color in there or 30% green color in there. So if we combine them or overlay them together, you will see the result on the right side. This is the color analysis result, shows the color distribution and color quantification result, and their ratio was automatically recorded. Wait a second, some people in here might ask why you choose the number 3 as the quantification numbers. I've been thinking the same questions. I don't know, because this is for testing. And later, I've been asked too many questions with this, so I thought there should be a way to find out the best number of level of greens if you want to analyze one image without human interference. And we have been testing, like putting the number of from 1 to 4, visually check what's the result look like. You can see on top level, you have different kind of green colors, and they are quantified in different numbers. And on the bottom line, you will see that their visualization in RGB 3D space. Their goal was try to find out how many colors are showing up and what's their locations and what's their distributions. Is there going to be more small colors show up, or is it all important for us to analyze that? So we do a maximum test, increase number from 1 to 20. Here I put them together, make an animation. You will see that on the left side, it's a 2D visualization of the color clustering changing from number 1 to number 20. And on the right side, you will see there are more colors from 1 cluster and until 20 clusters show up. When I see this, I'm so excited. There are so many small clusters show up on the RGB color space when we increase numbers. That means they are not so important. So we might find the best way to find out the opening number among 1 to 20. So here, we need to bring in a mathematical way to quantify them. In here, I mentioned about the elbow method to help us find out the best number of level of greenness you want to analyze. The basic idea was they calculate the within cluster, the distance, and find out the distributions and find out the best numbers. So in here, we are able to quantify the colors. We are able to find the best number of colors for the plant surface image. So I built up this smart pipeline. Smart pipeline, the uniqueness was it can compute both morphological and physiological traits. For example, the morphological traits, the width, height, and the number of leaves, that part, we also introduced a pre-trained model to help us segment the plant besides the dominant cluster algorithm. But I want to mention this, the uniqueness and the powerful part of SMART was the color analysis part. On this side, you will see the color analysis result I showed you before. With this SMART software, I was thinking, this is a software for plant science community, but what is the benefit if plant scientists want to find out more scientific research goals, or what's the scientific value for that? So I've been trying to read a lot of papers, and go back to my second slide, I mentioned about why the plants are green. I noticed the chlorophyll content. Chlorophyll content, they have some correlation with the RGB colors in some way. This result or conclusion was proposed long time ago, and recently some similar literature or papers try to do some research on that, confirm that too. So I'm very, very interested. How about let's try with the color analysis, then try to using RGB image only, then estimate the chlorophyll content. So the basic idea and the fundamental idea was using RGB images, then transfer them into another color space in computer vision part. Another color space was called LAB space. Then from LAB space, there are some direct connections with chlorophyll content. Why? Because if we look at on the right side, you will see that the RGB channels, if we convert them to LAB channels, L channels represent the brightness of the whole image, and A channels represent red to green, and B channels represent yellow to blue. So did I mention about the reflection in the beginning? Most of the green and yellow was reflected by the chlorophyll. And then, very, very interesting, LAB space, AB space captured the reflection part. So there some color information was encoded in RGB space, but if we transfer them into LAB space, this will be a big interesting thing in here. So we did some initial tests for compare with the commercialized devices to measure the chlorophyll content. On the left side, you will see this is a result from the palm chlorophyll flow meter, and the three circles, they represent where they measure the area and the related numbers indicating the readings. On the right side, you will see the color clustering result coming from SMART, and in the middle part, it is transferred from the RGB space into LAB space. For comparison, I put the palm result and the LAB color cluster result together. You can visually compare them. They are similar. That is very good news for us. So we developed the software and pipeline, try to using color analysis result to estimate the chlorophyll content. Thanks to our collaborators, they using the palm device, collect some data, and we try to use the color analysis result to compare with them, get some correlation. This is one of the results I'm going to show you. We did see some very good correlation here. So, but we can't rely on only one device. We need a multiple device to validate the result. So another collaborators, they are so good, they help us with using another device called Photosynthes Multius Q. This device, handheld device, to measure the chlorophyll content directly, and we use the SMART to generate color analysis result, compare with their result, get correlation. The result was consistent and good. From here, we are confirmed the SMART has the ability to estimate the chlorophyll content by transfer the RGB images into LAB space. So I've been continue thinking, because I learned from the plant scientists, the chlorophyll content has some connection or good correlation with the leaf surface or canopy temperature. Is it possible we still using the same technique or in here about the color analysis to estimate the temperatures of the leaf surface? So I've been digging out so many papers and find out they're supporting. I'm so happy to find out there was a very old paper and the recent paper. They are trying to do the similar things, but their result was limited into some specific plants. So I tried to do the very bold experiment. So we get some data from another collaborator of ours. They use the handheld infrared thermometers to measure the surface temperature of the leaves, and we use the SMART to generate the color analysis result compared to them, and we still very interesting. We got good result and good correlation. From here, I say that the foundation of SMART was the color analysis result and dominant color clustering method. And we are able to monitor the plant growth over time. And now the SMART pipeline was deployed into the high-performance PC and open source, free to use. Well, what you need was register a Cyverse account. And our SMART modules was integrated into our lab, the website called Plan IT. If you are interested, you can register and test it. The speed was very fast, but you need to wait in a long line because high-performance PC was busy. And on the right side, you will see a small tutorial teach you how to upload your own data and click the run to submit your job. And from that, I would like to thank all our collaborators because I learned from this conference at the first day from workshop from the Professor Katie. If you want to work fast, you work alone. If you want to work far away, we need to work together. So I'm really, really inspired by all our collaborators, and they are from different fields. Although I put a world map in here, most of them are US-based. And I'm really special thanks to the Syngenta friend. He's so interested in my pipeline, and he invited me to do some other tools. We built up a commercialized tool called Photosynthes, and another one from England research department. So my dream was let more people use it, and I hope the users will spread out into the world. Last one, I want to thank to our lab members and our mentors, and special thanks go to Jennifer Clark to offer me this chance to speak in here and offer me the trial grant, make it all possible. Thank you.
Thank you very much for the nice talk, Suxing. Is there any questions for him? Oh, just in the middle, that was fast.
Okay, thank you for the great talk. Well, as I also come from the computer science community and I also found the wonders of plants and learning a lot of biology, I really connected with the first part when you talk about photosynthesis. But then I got my first question that was quite fast is, why are you using RGB? Well, then you move to LAB color space, but isn't it more natural to think about using some like hue, saturation, and lighting because you can put in different dimensions the color, the saturation of the color, and the illumination, so you can forget about the illumination conditions and only do the clustering in the hue space?
Can I repeat your question? You are trying to ask me why I need to transfer RGB space into LEB space. Is that right?
But why don't you use some other cylindrical coordinates color space like hue, saturation, and value, or hue, saturation, and lighting?
Okay. There are many more color space in the computer vision part. LEB space was one of them. You are mentioning maybe HSV space, is that right? Yeah. Each space, color space, has their own uniqueness. But for plant images, I noticed most of plants are green color. We can say that safely they are yellow or purple. But the LAB space, as I mentioned before, A and B space capture the red, green, and yellow to other channels. That is what we are looking for and what we're expecting for. So using this as a cue, it is easy to capture the change of the colors. That is the foundation and basic idea in here.
So maybe it would be nice to look because the hue saturation, you have the color in one coordinate, that is the hue, that you can get all the colors there, and the saturation will say like how much green is that. It can be more or less green, but you can find it like in a big cluster of is this the color and then the amount of the color you have.
Yes, thank you. That's very good suggestions. I will try to look at that. And yes, thank you.
Thank you.
So is there any other question?
Yes, I had as well a suggestion for the HSV space and maybe nearest neighbors to get the clusters. But the other one is did you control for illumination using color checker to maybe improve the regression values?
Yeah, thank you. That's a very good question. Yesterday, I have listened to one question talking about the illumination change and to control the camera has same illumination, capture the same plant images. That is because this color analysis was sensitive to the illumination change. We have come up with two solutions. One, we build up the imaging protocol, try to get the constant illumination environment around the plant. Second, if you don't have that any images, you take at a different time, different sunlight time, or different illumination environment, we will do a group of all your data and get the average or mean value, find out the constant greenness in this data set, then use that as a reference. Then we calculate the distance between all the greenness against this base reference level. That's the current solution we are trying to use. And that is very good. Good, thank you.
Okay, so thank you again for this nice talk, Sushil. And also a big applause to all of our previous speakers. So thank you for the nice session. So the next one is our poster flashes. And I would like to start with James Burridge, then followed by Joshua Carpenter, Tessa, Durham Brooks, Heather Manching, and Juan Manuel Romero Barrios. Just line up and then give your flash talk. And press the big green in the middle to proceed.
Good morning. Thank you for inviting me to talk. So I'm a researcher at the University of Tokyo, and today I'm talking about a farmer-funded wheat research project in the north of Japan. And one of the things we're interested in, as you can see in the photo there, is lodging. So in order to develop new tools for these farmers that want to understand how to improve precision management, we're using a combination of ground truth data and a variety of UAV flights. So we have our different experimental treatments, which are fertilizations at different times, and we have a variety of ground truth measurements. And like I said, one of the things we're interested in is lodging and its connection to performance, well, its connection to growth dynamics. And one of the things we found is that lodging is obviously connected to height and topographical exposure in the field, so it's exposure to wind. But interestingly, the potentially problematic difference in height was visible in April. So that opens the opportunity for farmers to manage for height. Another one of the objectives or tasks that we're interested in is improving head density counts and also creating capacity to estimate head size profile. So we're collecting a variety of different scale data from CT scans to close range photographs to proximal photographs in the field in order to use a deep learning algorithm to help us estimate head size profile. And so with that, thanks. And if you have any questions, let me know.
Hello, my name is Joshua Carpenter from Purdue University, and when you want to quantify the root structure architecture of field-grown soybeans, you first need to consider the effect of the three-dimensionality of the architecture. You also need to know that when you go out to the field and you dig up these samples, the roots will be oriented strangely and they will be damaged by that excavation process. So these are two considerations that need to be taken into account. This work is demonstrating that you can easily and cheaply quantify take into account the three-dimensionality of the root system through photo scanning. And you can build 3D geometric models of the root systems. And these models have distinct advantages over 2D sampling methods. And they also can be used to quantify the effect of soils on the RSA. So if you look at panel A, you can see our photo scanning booth. We take a bunch of images, and those images can be used to create a 3D point cloud. We then developed a method for developing a hierarchical cylindrical model of the 3D point cloud. That's in panel B. C demonstrates the unique advantages of taking into account the three-dimensionality, namely that you can see the full distribution of a particular feature of interest, as opposed to sampling or 1D imagery that you would have to find you'd have to find major branches and you're just sampling. So you can see the full distribution. But we also wanted to know, how well can these models detect properties that are known to exist in the roots? And so panel D is looking at known properties of natural branching systems, specifically the law of area conservation, the self-similarity of the branching rate and the diameter. And we find that these models are able to quantify those known properties. And then finally, panel E, we look to see, can we detect, even though we know the samples are going to be damaged from the excavation process, are they still good enough to be able to detect the effects of the soil on the RSA? And here we show that we are able to detect the difference between in soybeans grown in a sandy soil versus soybeans grown in a clay soil, and different features change based on those soil types. So that's the research. If you have any questions, I'd love to talk to you later. Thank you.
Hello, are you wrong? Sorry. Oh, good. There's Tessa. Hi. The big read.
Okay. Hi, my name's Heather Manching. I'm a research associate out of North Carolina State University. I know all the posters have been taken down, so please scan the QR code for a copy of the poster if you want to reference it. My contact information is on there as well. But today I'm going to share with you a little bit about the research that we work on. And to give it a little bit of context, I'm part of an initiative called Breeding Insight On-Ramp, which it works with USDA breeding programs, specifically for specialty crops, and integrating advanced technologies, whether that's tools and software or breeding technologies into their programs. And so one of the branches of that research is focused on how can we bridge the gap between these awesome machine learning and computer vision algorithms and actually make them available for real-world breeding. This is definitely a challenge, and it's a huge gap. So a lot of my work is really hands-on, dirty in the field, getting to know these breeding programs and trying to figure out what is their breeding objective and how can we link it to something in the upcoming technology world to make their lives a little bit easier. So we serve kind of as a translator and say, okay, well, I have a breeding background. I can understand your objective is yield. What is the machine learning objective that could be used to accomplish that? So here is just a use case where yield is important in citrus. It's important in everything. But for citrus, it can be very time-consuming to measure, and a lot of specialty crop programs don't have the resources needed to be able to accomplish that. So here we're going to use object identification and counting to be able to estimate yield from images taken of trees from front and back. And so one of the major or most important concepts to developing machine learning algorithms is actually getting the data to train your models. And then after you have a model, you still need to be able to collect data to be able to use that tool. And so we integrate a lot of digital data collection practices. One of our main tools is Fieldbook. Please scan the QR code if you're interested. It's an open source Android application for phenotyping in the field. One of the cool things about Fieldbook is you can actually take image traits. So you can take pictures directly in the app. It will automatically assign names to your images, so you don't have to go back and rename them all after the fact. But we use this digital data collection technique to collect images of citrus trees. We have a publication now for CitDet. Again, I love QR codes. Please scan the QR code. This data set is publicly available. If you scan the QR code, it'll take you to a GitHub page, which goes over the whole project, as well as all the annotations and the models that are present in that paper. So we created this digital data collection of images. We want to be able to share it, which is why we made it publicly available, because we want other people to come in and use this model and use this data set to train models that can accomplish similar tasks in their programs. The next step, and I loved Addie's talk, is model development and validation, and this really revolves around transdisciplinary collaborations. So we've worked with University of Texas Arlington to develop this model. And then the validation, we need to show the breeders that it actually works. So comparing it to ground truth data, we found that there's less than a 1.5% error rate in estimating yield. And in terms of breeding, we can capture 4 out of the 5 top ranking trees based off this model. Integration, what are the logistics of a breeding program? They're complicated and time consuming and very strict schedules. So we need to show that we can save time. And if we compare the results between harvested counts and machine learning estimate counts, we can save about 73 person hours to be able to accomplish the same thing. And then finally, deployment. So what do we need to do to be able to deploy these models? We need to take this model and optimize it for low power devices. And then there's a publication in process here called Mobile FruitNet, where we've done that with this model specifically, but we're also working at other ways to deploy apps that are available and can be used by real-world breeders. So with that, thank you.
Hello, I'm Juan. I'm from Argentina, and I'm working nowadays as a postdoc in Jülich, Germany. So we know for sure that CO2 concentration will go up in the future and that we will need to produce more food. So it's reasonable to ask ourselves, how will these CO2 elevated levels will affect crop production yields, plant pathogen interactions, and also the photosynthetic efficiencies. So in Jülich, we have this amazing facility which allows us to enrich the CO2 concentration in the atmosphere in the field. It's called free air CO2 enrichment. And through this semi-automated positioning system, which is called FieldSnake, we can take measurements of both active and passive fluorescence of the plots. This active and passive fluorescence approach, a combined approach, allows us to register at the same time the photosynthetic efficiency through the lift device, which is an active methodology that irradiates the plants with blue light and registers red fluorescence. And also through the Flux device, which includes 2 different spectrometers, we can retrieve sun-induced fluorescence in a very reliable way. So we have 2 experiments where we have more, I'm showing 2. Sugar beet experiments where we test 2 genotypes with 2 disease treatments and in 2 CO2 treatment. That's ambient CO2 concentration and elevated CO2 concentrations, which is around 600 ppm. And also we have a wheat experiment with 10 genotypes that were relevant to the farmers around the area, also growing in the ambient and elevated CO2 concentrations. So briefly, if I can point some results, I can say that the combined approach that we developed allows us to disentangle the physiology and the structural effects of the canopy on the SIF and the PS2 efficiency measurements. And last, very interestingly, elevated CO2 concentrations cause the photosynthetic efficiency to go down instead of up, which is kind of counterintuitive, because if plants are happier, why they don't have a higher photosynthetic efficiency? So we think that it's a kind of feedback inhibition. Also, this is quite expected. Elevated CO2 cause an increase in the beet and the grain yield. and also it's kind of make the sugar beet plant less vulnerable to the disease. So that's all. I will invite you to discuss this in poster 96, but poster 96 is folded there. So if you want to talk about this, you can ask me. Thank you.
So now we're almost at the end of the very last session. Thanks for staying still here. But we still have the time to again ask questions to our keynote speaker, Theo Vandely. So if there are any further questions about disease detection and how to integrate them into breeding programs, feel free to ask. Sorry. Any question left? Not anymore. Oh, back there. Okay.
Well, great talk. Just a question about, these assays that you must perform, they're mostly manually. And what would be the challenge to bring this now to high throughput so we can really make sense of the complex biology behind that?
Yeah, indeed, this is quite a hard labor sometimes to do all these inoculations. We've automated some of them. In the greenhouse, for instance, we are able to automatically inoculate the plants, for example, monos, so that helps a little bit in the throughput. But in the field, there's also possibilities, I think, to automatically inoculate, at least with the traits. So there is potential, but it depends a little bit on the topic of study. Because if you have obligate biotrophic fungi that I also showed, it can be hard to get enough inoculum to do this automatically, because you need a little bit higher volume, and then it gets more complicated, and it's back to the handwork. So our key thing is actually to work on the processes, the mode of action, in a small scale experiment, and then implement that in the larger scale when we know the mechanism. And hopefully then it will not, if we know the genes, if we even can transform the genes into the crop, it will be it's just a validation that you have to do in the field. That is the approach that we try to follow. Does that answer your question? Yeah.
Is there another question? Up.
Thanks, Theo, for your speech. I mean, beside the sensing of the disease and the spread of the disease, I mean, are there a kind of mechanistic models that can help you to describe the dynamics in space and time at that scale of the disease? And how do you see the link between the observations that you get from your high throughput phenotyping experiments in such kind of models?
Yeah, that's a very nice question. And well, obviously there's other factors that we consider. I already mentioned, so is the plant, the age of the leaves, things like that. So if you have crop growth models, for instance, that can help, I think, to get a better grip on this disease expression. So we are working on these crop growth models in some of our crops, and that could be implemented perhaps in a later stage. We have, in this case, there's a lot of challenges, mainly because you see so much genetic variation, and I didn't hear that much in most of the talks, but there can be substantial differences between genotypes, and that's something that you have to take into account as well. Hopefully in the future there will also be more information on the modeling of 3D, for instance, for the different genotypes, and implement different growth models for the different genotypes, and then perhaps that can be it can be integrated, but it takes time. I would say.
And eventually can be introduced in such kind of models the different susceptibility of the genotypes in the propagation at the leaf, but also at the canopy scale of the diseases? Do you think it's possible using high-throughput phenotyping in control conditions? Or
yeah, so if you have that information available in your metadata, I think it can be used. It's well known actually from literature that some of these infection processes, for instance in the botrytis, can happen quite early in the stage, like for instance in the flower, but then there's a long latent period until fruits start to develop and things like that. So there are specific processes that occur during the development of plants, and you have to take that into account. If you have different genotypes, perhaps you will have earlier development of fruits, and then also the disease will be impacted by that. So also the hormone developments. Yeah. So there are a lot of things connected. At the moment, I'm mostly focusing on the disease, capturing really good images, high quality images of the disease process, and trying to find the genes that are responsible for the resistance and susceptibility. And the next step, hopefully it will follow maybe with this new machine learning and AI type of technologies. But I think it surely will depend also on high quality data to start off with, and the literature that is also available. Yep, thank you.
So, is there any?
Hi, it was a great talk. On the first day here, I heard that X-ray imaging can damage the plant, and now I heard you say that taking pictures can also affect the growth and the diseases on the plants. What can you tell me more about it? Some insights? Is it because of the illumination, or why did you say that in particular?
So if I get your question right, it's about the 3D structure and the impact on the disease? Or
no, it's about the imaging activity the plants can affect both the growth or the disease spreading.
Yeah, that's a good question. Yeah, so the light actually has a lot of effect on the plant, and the measurements that we do actually we saturate the plant with light in order to capture the photosynthesis. So we wanted to do some cool movies, obviously showing the effect on the progression of the disease, and then we found out that actually it was quite remarkable that we had delay of disease expression in these plants. Luckily, my PhD student also had control samples which were not exposed and that were actually in similar conditions all time, and we saw a pronounced effect actually on the disease development. So that's why we started to go a little bit lower resolution, a little bit higher distance, and we are measuring a little bit less frequently in order to still have, let's say, the similar disease progress compared to our non-measured control plants. But it was a pronounced effect, and I think, there are also people interested in this, because perhaps with the light, changing the light condition or with flashes, it will be possible to reduce the infection and the disease progress.
Oh. Any further question? And maybe from my side, so how far do you think are we to not just detect that there is a disease, also in the field, and know what kind of disease it is?
Yeah, that's a difficult question, because it depends really on the crop. I think in most cases, most of the crops are in particular region are usually affected by the same disease. And sometimes it switches a little bit after a few years. You can maybe have something new in. But if you have, for instance, potato, you will likely have potato late blight in the end of the season. So many things are already known. And there may be some curious disease that we don't have enough images for to really call it in this way, but probably they're also not the most important one. I think it's important to separate, let's say, the top 5 or top 10 diseases per crop, and then depending on the season, depending on the symptoms expression as I showed, and depending maybe on the resistance that you have in your crop, and maybe spray use, I think it will be possible to capture those quite well. But there can also always be new things, and and then you have to start again. Yeah, but we noticed actually we have inoculated our plots, and we noticed actually some other diseases that came into our plots. Yeah, we could directly tell this is not, it looks really irregular, so we could directly tell this is not the inoculum, this is something else.
Yep, thank you. So if there's no question left, actually, I'm happy to thank you again. All the speakers of the session. And we'll give over to Jennifer.
Yeah, got it. Okay. All right. First of all, I wanted to say thank you to everyone here. These conferences and these events, and the great science that we saw and we heard, and the posters, that none of this happens without you. We talk about the network, but it's really the people. So I want to thank everybody who attended. Second, anyone who is a member of the incoming network board, could you please come up here? I don't know who actually is. Alex. I see Alex. Rick can come up because he's our new treasurer. Vincent. You forgot. That was reminder number 1. Okay. So I also wanted to thank is Lee here? He might be wrapping up his booth.
I was.
I wanted to make sure that you guys know the people that are involved with the IPPN board. So if you have questions or suggestions, questions and suggestions you can send to me. Complaints you can send to them. But this is your network, right? So we love to hear from the community. We love that your engagement with the working groups. And I think that, speaking for the board, we would love to have everyone to enable collaboration and to highlight the discoveries and the advancements in the community. So if you have news, suggestions, exciting things happening, please let us know. We'll be sure to include that in the newsletter. I wanted to give you a preview that next year there will be a plant phenotyping conference hosted by the EPPN. I'm looking at Philip so he can confirm. Emphasis? DPPN. Okay. The European thing. Yeah. And the IPPN General Assembly will be held at that conference. So there'll be additional news coming, but just very early preview that in 2025, there will be a phenotyping conference in Europe, and we will host the IPPN board meeting at that event. Okay. Any comments from the board members?
I could
go for it.
Yeah, so I've been in the previous board, and now I'm only the treasurer, so that's a small task. But I've also been in the imaging working group, which was called differently. It's now morphed into the AI working group, you could say. I just want to make you aware that there are many different working groups, you know, but you could join actively in discussions if you find the topics of the working group relevant for your research. And if you then look at these working groups and you can't find any topic that interests you, you could launch your own working group on a topic because topics change. There's things that were relevant 8 years ago are not relevant anymore today. So if there's something missing, satellite stuff, I'm just suggesting something. With a lot of interest in your talk. So we could create working groups just to make you aware of it. So that's it.
Right. Yes.
Yeah, and I would like to thank this to take this opportunity to call to the front also all the network representatives from Australia, from the APPN, Richard Dickman and Valatio, from the NAPPN, also Roland Pirushka is a representative of the German European Plant Phenotyping Network. Please come on with us to the front. Who did I forget? Stein, emphasis director, Jacob Jess from the APPN, please come to the front.
Their APP.
That Austrian APPN. The Wei Guo from the Japanese Plant Phenotyping Network. Wei, please come to the front. Ready? Who did I forget? John from the South African Plant Phenotype. Please come here. Yes, John, I know you don't like it, but we're trying to get all the faces here that all the people can see, in terms of networking. That's John. Who are the responsible people? Mr. Kim from the Korean Society of Plant Image Science, one of our new partners, please come on to the front. And I'd like someone to take a picture and get that to me, please. That would be highly, highly appreciated. So did I forget someone? No? So the idea of our network, especially on our International Plant Phenotyping Network, was that we are facing and trying to tackle global challenges. And these challenges often do not direct themselves along border lines, not national, not regional ones, and let this simply be a symbol that we are all pulling together on one string in the space of plant phenotyping. Thanks.
All right. Thank you. Entering at the time. There you go. Those mentioned.
Okay, I think I have 3 more announcements. One was that even though he couldn't make it to this event, I wanted to extend my gratitude and thanks to Uli Schur, who was the chair of the IPPN for a long time and put considerable effort into building the IPPN to what it is today, represented by these people on stage, right? So we owe a big debt of gratitude to Uli. And so I wanted to make sure that he was recognized among our community. So big thanks to Uli. Okay, next I wanted to invite Gregoire to come to the stage. Yes, you guys can. Thank you very much. Yes, you may. So as those of you who attended the banquet last night would recall, we had a raffle of the Photosynth device, and the winner was Phenospec's. And so very graciously, I want Gregoire to describe what we would like to do, but I think this is an excellent opportunity for collaboration between academia and industry. So,
yeah.
Please.
Yeah, thank you. So I'm Grégoire Hummel from Phenospec's. We won yesterday this super nice prize. I was super happy. And because I really wanted to have it, I wanted to do some nice experiments to compare the data of the plant eye of our instrument with this instrument, because often customers are asking us, hey, Greg, how is this instrument, the plant eye instrument, and compare the data to a spot, for instance. And in order to better consult our customers, I said, okay, cool, let's get that thing, yeah? So I sent my entire team to the booth, and everybody should take care of the lottery. And then we were lucky, we won the thing, right? I was like, whoa. The thing is, and afterwards we thought, maybe it's not so nice, right? I mean, it's a super nice initiative here, and we are a private company, like this is rather maybe for PhD students or master students. They should profit from this initiative. So we said, okay, maybe we should donate this thing and give this actually to university, which can make use out of it. Right? And that's what we would like to do. And on top of that, we want also to add one of our instruments, a microscan with a plant eye that if the student that you somehow will select is interesting to make this validation study, so we can also provide this tool, we're going to ship it. And and if there's nice data coming out, maybe in 2 years, when we meet again, the data can be presented at this conference. So yeah, thank you. So I give it over to you. Take care of it.
I'm gonna get sick.
Yeah, thank you. Bye, please.
Thank you very much. Thank you. I thought that was extremely gracious, and I know a student will be very, very happy. So lovely. Thank you, Gregoire. Okay, right before we close, last minute housekeeping items. There will be boxed lunches provided. So whether you're going on a tour or you're hungry, great. There will be boxed lunches out front where we had the coffee breaks and where we had lunches. So there will be boxed lunches for you to take with you. There are 3 tours coming up. So there's a tour of the Greenhouse Innovation Complex, which was close to the opening event, if you attended the opening event. There is a tour to our field facility, our field, I don't want to call it a gantry system, a suspended camera system. Spider cam, thank you. Spider cam system, that's one tour. And then there is a tour to LICOR, which produces photosynthesis measurement equipment. So if you have signed up for one of those tours, there will be buses leaving from the front area. So if you go to where the coffee break was, we'll gather everyone and get you guys on your tour buses. Okay. I think that's it. So big thank you to everyone. And I hope to see everyone, if not next year, then in 2 years when the next IPPS will be held in Africa. All right. Wonderful. Thank you all.